Hi, this audio recording was created to allow me to finish the remaining material in the lecture I gave on March 2nd, which was focused on genetic studies in diverse populations. And we're going to start on slide 57, which is where I left off. Slide 57 shows sample sizes needed for doing admixture mapping studies. And if you take a look, for example, at the first or the top half of the table, where allele frequency difference of 60% is equivalent to delta, which I defined in class, which is the measure that allows you to define an ancestry informative marker. And you may recall that I said anything greater than 30% difference between two populations would be considered an ancestry informative marker. So this table takes a look at three different allele frequency differences between two populations, African and European. So looking at 60% as one example and the different combinations that would give you a 60% difference, you can see that for the allelic relative risk, which is comparable to the allelic odds ratio, which we've been talking about in class, as that goes up, the sample size becomes smaller. And this is shown, for example, allelic relative risk of 1.5 on the first line. You would need 1,418 families if the SNP that you were looking at had allele frequency difference of 60%. One was 70% and one was 10% in the two populations respectively. So slide 58 shows a graphic representation of results that you can get from the LAMP algorithm. So the LAMP algorithm is used to do local ancestry analyses. In fact, we use this method in my own lab. And what this figure shows is the assignment locally of ancestry throughout the different regions of the genome. And you can see for this analysis, the three populations that were characterized, European, Native American, and African, suggest this is showing data from a Hispanic population. And you can see by chromosome the different regions of ancestry as you move along the genome. So this analysis can be very useful if you're trying to look at candidate regions in the genome or just doing a whole admixture mapping. You can see if you had a group of individuals that were cases and controls that you could look at specific regions of the genome for ancestry differences. So this is just one of many methods, actually, that are available now to do these kinds of analyses with genetic data. So slide 59 talks about population stratification, and I just want to define it and then talk a little bit about it. So this is a primary consideration in genetic epidemiology studies, and it's such an important topic because any publishing that one does now of genetic studies, case control association studies specifically, have to address this potential source of confounding. And there are multiple ways to approach this problem, which I'm going to describe. Another way of thinking about population stratification is population structure. But typically, population stratification is used to refer to the possibility of genetic ancestry differences in cases and controls and the confounding that might result in an association with a particular SNP allele. So let me give you some examples. Moving to slide 60. So the true definition of population stratification is as follows. that differences in allele frequencies between cases and controls are observed because they're really due to systematic differences in ancestry rather than a true association of a gene or genes with a disease outcome. Another way that population stratification is defined is that it's a distortion of an observed association between a genetic variant and disease, and it occurs when a genetic variant is associated with a true risk factor that varies by race ethnicity. It's a type of confounding, and studies who use admixed populations are susceptible. But let me just emphasize that basically every study, every population that you might examine in a genetic study has some level of admixture. And there is the potential for population stratification in any genetic study. So here is an example showing, first of all, for population 1, the results from allele counts in a case control study. And if you look at the counts and convert those to proportions, you'll see that the proportion of the A allele is nearly identical in cases compared to controls. And so for population 1, on slide 61, there is no evidence of association between the allele and the phenotype. And if you look at population 2, you will notice that the proportion of the alleles in the cases and controls are also similar. But if you notice, the allele frequencies are very different in these two populations. And if you look at the frequency of allele A in population 1 and the frequency of allele A in population 2, you can see right away that they're very different from one another. And if you look specifically in the cases, you will see that the frequency of allele A or B, but let's just focus on A, is different in population 1 compared to population 2. So if you pool those two populations and then ask about whether or not there is an association with allele A and phenotype, you will see that there is evidence now for an association, but it's a false positive association because it's entirely due to the fact that there's population stratification. There is confounding by genetic ancestry here, and that is exactly the type of problem that we're trying to address in genetic studies. This is an extreme example, but the bottom line is that this is a very simple example of what we're trying to get at when we do genetic studies, and we want to make sure that the association we observe is real. So this is just a DAG showing that for example, case and control allele frequency differences, as I showed you earlier, are due to systematic differences in ancestry rather than association with disease. So you can see from this graph that genotype is confounded with race ethnicity. And so if you are going to do a genetic study in cases and controls, you have to, first of all, identify the evidence for population stratification, and then you have to actually use some sort of method to adjust for it. And that way you can account for the potential of confounding and accurately interpret your results. So population stratification, I'm on slide 63, occurs under the following three conditions, and this will help clarify what I'm trying to say. So the first condition is that the allele frequencies have to differ between subpopulations. So like what I showed you earlier between the two different populations, the allele frequencies were very different. And disease incidence rates need to vary across subpopulations, such that the potential for your cases, as one example, might be to have a different proportion of individuals who are European than controls. So those two conditions have to be satisfied. And then a third condition that needs to be satisfied is that the genotype frequencies and the disease rates must vary together. So to think about that in a simple term, you could have a disease like multiple sclerosis, for one example, and it tends to occur in individuals who are of Northern European descent. So the genetic ancestry is likely to be of Northern European. So you could imagine that if you were doing a case control study and your study participants, even though they identified as white, non-Hispanic, the cases might be likely to be much more European. They might have much more Northern European genetic ancestry in them compared to controls, just because they happen to be cases, and that's the highest risk group. Well, I should say it's one of the highest risk groups. It's more common in Northern Europeans. So if you happen to look at a genetic marker that also has a higher frequency in Northern Europeans, and you looked at that genetic marker in cases and controls, you might see an association with that marker and disease just because it's correlated strongly with Northern European ancestry, and your cases happen to be of higher proportion of Northern European ancestry. So that is what this means in 63 and in 62. What I just told you is shown in the diagram. So I'm moving to slide 64, and which shows some solutions to this problem. So the first one, obviously, is to use the transmission disequilibrium test, family-based test for association. Remember, it's a test of association in the presence of linkage. And the reason why that statistical test was so popular for so long is because it did greatly eliminate the possibility of population stratification contributing to a study. So the idea that you could get false positive associations was greatly reduced by using a family-based test like the TDT. You can also match your controls and cases as best you can, and for a very long time that was done using self-reported race ethnicity. And even though self-reported race ethnicity is very highly correlated with continental genetic ancestry, it may not always be accurate, and the effects may be much more subtle, the genetic differences, genetic ancestry differences, than just sort of larger continental assignments. So we need better methods than these two solutions. So slide 65 shows just a few of a lot of different approaches that have been developed over the years. And I'm going to talk about some of those approaches just to give you an idea of what's out there. And also when you read papers, you'll have a better sense of the approaches that are being used. So genomic control, I'll show you, that's been around for quite a while, and it attempts to correct overdispersion of test statistics due to stratification. So I'll show you how that works. Maximum likelihood method has been used to estimate individual admixture measures, and then you can use those actually as covariates in a model. And then there are other methods, structured assessment, which is similar in terms of the final approach that's used, which basically is adjusting for genetic ancestry as maximum likelihood method. Just different approaches, though. maximum likelihood versus a Bayesian method. And then principal components analysis, which captures genetic background as covariates, which can also then be used in models for adjustment. And then multidimensional scaling, which is similar to identity by state, which we talked about, which is a measure of sharing. It's a measure of allele sharing. And that is commonly used, large scale, to get a sense of how people are sharing genetic or genotypes across the genome and how closely they're related or not. So moving to slide 66, I'll first talk about the genomic control, which is a non-parametric method that's used to control for population stratification in a case control study. So if you're comparing cases and controls, and they are different, even subtle differences, with respect to genetic ancestry, you could have an inflated null distribution of the chi-square statistic. And so what you can do with genomic control is that you can take a sample of random loci. So this assumes that you're doing lots of genetic tests across the genome. So you could just take a set of those, assuming that they're just randomly selected and not associated between disease or between case with the allele and case status. So you're just taking a random set, and you can use that random set to estimate a variance inflation factor, and I'll show you how that works. And then you can use that lambda, the variance inflation factor, to adjust then your observed chi-square statistic. And this is a method that's been used for a while. I'll say a little bit more about it when we go to the next slide, but you can use it to control for spurious associations caused by population substructure. And it's an okay method. It's actually usually used now largely kind of as a QC measure, but it can under or overcorrect, and I'll kind of show you what happens with that. So I'm going to slide 67 to give you a little bit more detail. So if you take a look at the definition of lambda, so the lambda that you're trying to estimate equals the median of your randomly selected loci that for which you do a case control comparison and generate a chi-square statistic. So you take that group of chi-square statistics that you generated from case control comparison and take the median of that and divide the median by what would be expected, which is 0.456. That's the expected median value for chi-square statistic for the null distribution. So if you can see that if you take what's observed, the median of what you see, and if there is no inflation, no population stratification in your study, then that median value should be pretty close, if not 0.456. So lambda would then be 1 or very close to 1. But that's not maybe what you would see if there really was something going on. So maybe it's a lambda then can be so if the median then is a much bigger number than 0.456, the lambda value is going to be greater than 1, and that's what you're looking for. You're looking for what to determine whether or not there is a lambda that's greater than 1, and then you can use that number to correct for your observed chi-square distribution, and then that would give you the new chi-squared distribution. And so you could use that for all the chi-square tests you do across the genome, or if you were only interested in one candidate region or something like that, then you could adjust just that region. But basically, if the inflation factor lambda is greater than 1, it indicates that there's population structure or it could be genotyping error. That's why I mentioned it's used largely now for just a quality control, just to kind of like Hardy-Weinberg equilibrium, just testing to see whether there's something really unusual going on with your data. But you can carry out an adjusted test of association using lambda that would take account of mismatching of cases and controls at any particular SNP of interest using the statistic that you generate from it, the chi-square value, divided by the lambda that you calculate from those randomly selected markers. Now, another way that you can look at whether or not there's evidence for population stratification in your study is to plot all your expected chi-square values, and that's just the under the null, so that would be a straight line, and then you can plot your observed on that same plot, which in slide 68 this is called a QQ plot, or a quantile plot. And if you take a look at slide 68, you will see that the null distribution is the straight line that's plotted for the expected value. And then you can see that before adjustment, in the darker purple, you've plotted your chi-square values from your study, and then the adjusted would be what you would plot at what you would plot what you would calculate after you divided all of those by your lambda value. That would be adjusted. And what you see is that the dark purple, it deviates more from the expected under the null, and the gray, where you've adjusted, brings it closer to the null, and you can then assume that those plotted chi-square values for the SNPs that correspond to the light purple dots are likely to be true associations. So whatever those SNPs are, they are probably real associations with your case status. So I'm moving to slide 69, and here's just a description of doing a structured association test, and this uses ancestry informative markers to estimate individual genetic admixture values, and then you can include those admixture values as variables in a regression analysis. So remember that those ancestral proportions add up to 1, and so they would be continuous measures. And for example, if you want to include African admixture as a variable in a regression analysis, then you would just use every single individual's proportion measure that you estimated using ancestry informative markers. Now, you can, as I mentioned this earlier, you can get pretty reliable estimates from even just a small number of SNPs, like 100, to adjust your models. But what happens is the more homogeneous your populations are. So if you are working with European populations, they are more homogeneous than more recently admixed populations like African Americans or Hispanics, and therefore you need more markers to get better ancestral estimates for adjustment. Okay, so moving to slide 70, this is just a description of how principal components analysis can be used in a genetic study to allow you to correct for population stratification. So again, everything that I'm talking about really relates to case control studies, because that's what's used now, has been used now for many years. And so principal components analysis is a very standard approach. So you would have your genome data, whole genome chip data, for example, generated for your cases and controls, and you would generate principal components using those data, and then you can plot them And then you can see easily in a plot, which I'm going to show you some plots, where individuals are outliers. So you can remove individuals who are really very far out in terms of not clustering with the rest of your sample. So that suggests that they're not not part of the more homogeneous sample. And then you can also use those principal components as covariates in your analysis. So similar to what I mentioned in the previous slide, where you use ancestry informative markers to calculate genetic ancestry estimates, you can also use principal components, because they're also individual measures, as covariates in an analytical model. Now, they do not give you estimates of individual admixture the way ancestry informative markers will, but they will give you continuous measures that can be used as proxies for population differences, and you can adjust for those in your model by just including them as a variable. And Eigenstrat is one common software that is used to do this, but the generation of principal components now is such a standard method. It's done in many software packages, and you can even do it with R packages. And this is just a variable that you generate for all the individuals in your study and then use it as a covariate in your models. Okay, on slide 71, there's a formal definition of principal components analysis. So it's a statistical technique for determining the key variables in a multi-dimensional data set, like a data set that we're using all the time for case control studies, which is this big genotype data set, whole genome SNP data. And you can use the principal components analysis to visualize the individuals in your data set based on their genetic information. And you can create these components, and it is the first principal component that has the largest variance. And so that one and a few others usually get included in a genetic model because they capture most of the variation due to genetic differences. All right, if you go to slide 72, this is an example. So this is substructure within West Africa. And what this slide shows is a plot of principal components that were generated from whole genome data derived from many different tribes in West Africa, so from different geographic regions. So if you look at the bottom, you'll see the key by color where these individuals are coming from. And if you look at the first plot, principal component 1 versus 2, you'll see how the populations fall out into clusters, and it's the first principal component that you see explains the largest portion of the variance. And if you look, you'll see that the populations cluster just the way they cluster geographically. So they're in separate groups by region, and they were also clustering by those groups based on genetic data. And those data, so I'm moving now to slide 73, and which shows a more recent paper that examined genetic data and geography within Europe. So, on slide 72, we're talking about West Africa. In slide 73, we're introducing the idea of admixture in European populations. So if you go to slide 74, you will see one of the most famous studies that was done a few years back, where a lot of different European populations were genotyped, and 197,000 SNPs from a 500K SNP array were used to generate this plot. And there are 1,387 individuals here, and principal components analysis was used to plot the different populations and by color. And if you look, you will see that there is a lot of evidence for this European substructure in this larger group. So here's a whole group of Europeans, which you might think is a fairly homogeneous population, and it certainly is if you compare it to more recently admixed populations. But with enough genetic data, note that we had to use a lot of genetic data here to separate these European populations. But if you look at them closely, you will see that they really resemble the geographic map, same as the previous slide. You'll see they cluster into the regions from which they came. And slide 75 just describes what I just told you, gives you the figure legend. So you can take a look at that, and it will help you interpret what is shown in slide 74. So a few other studies that are worth noting. Slide 76 is a paper from about 10 years ago that was the first paper to demonstrate this. So this paper actually came out before the Nature 2008 paper that I just showed you, demonstrating European substructure. So keep that in mind. This paper in slide 76 was the first paper to demonstrate population stratification in a European study. So it was a case control study that was looking at extreme ends of the distribution for adult height. And everybody that was in the study was self-described. They were born in the U.S. and self-described white or Caucasian. And what they found in this study was that there was a genetic association, but when they looked closely, they figured out that this LCT gene, a SNP in that gene, that SNP in that gene actually vary widely in the frequency across Europe. And it was associated with height, but it turned out that the cases, which were one end of the distribution of height and the controls were the other distribution, were actually not matched well at all for genetic ancestry. So the result that was observed here was due completely to the fact that there were different proportions of European ancestry in the cases and controls. And when they matched them much more closely, the association went away. And this was kind of a shock to people because up until this time, there was some consensus in the scientific community that European studies, white non-Hispanic studies, weren't that prone to this phenomenon of population stratification. And it was realized through this study and many others that followed that it was definitely a big issue that had to be considered even when you think you're studying a population that's more homogeneous, that there's very, very fine stratification. And if you take a look at slide 77, what this study, which followed not long after the study I just showed you in slide 76, what this study showed basically was that you needed a lot more genetic markers to pick up on population, the potential for population stratification in European studies, and that if you increased your sample size, that the potential for false positive associations or stratification causing false positive associations increased. So it was sort of counterintuitive. People, I think, thought, oh, if the study is large enough, then population stratification won't be an issue. And actually, what we learned was that the bigger the study got, the more important it was to think about the effects of population stratification. So more markers needed, and especially if studies are large, you're going to pick up even very, very, very small small evidence of population stratification. Okay, so slide 78 just is a plot, a newer study showing continental assignment based on genome-wide profiles. And prior to this study, this sort of similar study had been done using a very small number of, it was like 318 or so microsatellite markers. And what this study showed was that you could then go and use a lot more SNP data and get really good estimates of population structure. So you remember how these plots work. Every vertical line is a person, and you can see that if you take a look at the ancestral groups that correspond to the plot below, they closely match what's in the structure plot. This is from a program called Structure. And they cluster according to continent. And then you can see there's some evidence of admixture in, say, the Middle East. But these populations look fairly homogeneous, and they're assigned based on geographic region. Okay, a few last slides. I threw slide 79 in there just to kind of show you that this is a slide from that shows plots from a study that I've been working on. This was some time ago now, but what's here are individuals that have multiple sclerosis and controls, so cases and controls from a study that we've been doing at Kaiser, and they're on the left, so Kaiser Permanente, Northern California region, cases and controls. And then on the right, the very right, are the Ceph samples, Ceph data that you've heard about, so the CEU are the Ceph Europeans, and then Chinese, Japanese, and Yoruban. So the 4 different Ceph populations, and the original HapMap samples. And this plot was generated using Structure. Again, this is an algorithm that uses a Bayesian approach to assign genetic ancestry. And so this was based on using just 112 ancestry informative markers, so a small amount of genetic data. And if you look at how the plot looks if you tell Structure to use 2 populations, you can see that the Kaiser samples and the Ceph European samples, because the Kaiser samples are all white, non-Hispanic, fall into the same population, and then the Chinese, Japanese look pretty similar, and the Yorubans then fall out. Now, if you ask Structure to assign 3 populations, then you see Kaiser and the Ceph Europeans are still together, the Chinese, Japanese are together, and the Yorubans are just much more distinct now from the Chinese and Japanese. And if you then say, okay, let's assume 4 populations and try to fit genetic ancestry to these groups, now you see for the first time that the Kaiser samples, so these are just, as I said, cases and controls recruited in Northern California, so white Californians, they are actually different from the Ceph Europeans, and that's not unexpected because the Ceph Europeans are much more homogeneous in terms of European ancestry. So you can actually separate them. There's more admixture, European admixture in the Kaiser samples than there is in the very homogeneous Ceph European population. Okay, so the next slide, I think we're slide 80. This is just an updated list of the different software and methods that are out there to do the work that I've been describing. And if you were going to take the lab portion, computer lab portion of our course, you would actually learn how to use a few of these tools. So structure's listed there as the first one. You can get global ancestry estimates, and Structure now has a new software called Fast Structure, which we use routinely in my lab. And then there are others that are listed here. I'll just say that Plink, which is listed further down, is probably the software we use the most. That's a genetic analysis software that has lots of different tools in it. In fact, it's a big part of the computer lab that we teach. And then there are others that are listed here. LAMP, I mentioned earlier. You can get the local ancestry estimates as well as global. But this just gives you an idea that there are a lot of methods out there. There are others that aren't even listed here. And there are some like RF Mix and others that use random forests and supervised machine learning. Those are other methods that we're using in my lab for different projects. But this just gives you an idea that there are a lot of different methods out there. And then finally, slide 81, you can take a look at this, just takes you through a few of the methods that I mentioned that can be used to address case control studies and dealing with genetic ancestry estimates and identifying evidence for population differences. So identical by state, which is sharing. So that's A. You can see that. So IBS is just showing you basically comparing all the different subjects, all the pairwise comparisons that you would do in your data set for every single SNP, and you would ask about sharing 0, 1, and 2, and then you can calculate a distance matrix. And it's very similar actually to D, which is a data matrix that is generated also using IBS distance. And then D is not so different from principal component analysis, which is based on variance. So that's a different. C and D are similar actually, but they give you different. They're based on a different algorithm. Each of them has a different algorithm, but the end result are components that allow you to assign individuals to clusters. And then there is the Cochran-Mantel-Haenszel test, and that's more if you want to actually stratify your populations. That would be based on an a priori designation of strata. And it's how you could do an association test when you know you've got different populations. So these are all sort of different ways of approaching studies in genetically diverse populations, which I would argue are basically all population studies, because hopefully you can see that even in European populations where you would expect genetic ancestry to be more homogeneous, there is diversity, and there's the potential for population stratification in a case control study. And it's just much, much more pronounced in more recently admixed populations where you have, big regions of the genome that you can assign to one ancestry versus the other. And so there need to be different approaches for those populations. Okay, so last slide 82, I just want to summarize things that are really important about this lecture. And we talked previously about genome-wide association studies, and in this lecture I talked about the value of imputation for genome-wide association studies and the use of the 1,000 Genomes Project as one reference for imputation. There are others. I defined admixture, and I also described how it could be used to find disease genes and how it can lead to population stratification, which is a type of confounding that has to do with different ancestral proportions in your cases and controls, different incidence of disease in your population and a genetic marker that you're testing that has different frequencies related to ancestral differences in your population. And the relevance of population stratification to genetic epi, that is a huge thing that must be addressed, and there are many ways that that can be done. How to detect it, I've talked about that. And advantages and disadvantages to using self-reported race ethnicity versus genetic ancestry. Remember, those are two correlated but different constructs that capture different things. So they're both important in a genetic study, and it's more the interpretation of what those constructs capture that is something to remember. And then how population stratification specifically is addressed in a genome-wide association study. Well, you can use the lambda method that I described, which is based on unlinked markers and comparing the median of chi-square tests derived from cases and controls using those randomly selected unlinked markers to the expected distribution of the median of the chi-square under the null. And that should be 1 if there's nothing going on, but typically there is. And so that number would be greater than 1, and then you could adjust all your tests of association with that lambda value. That's one way that's been around for quite some time. And the other ways that I described include determining genetic ancestry proportions and using those, because those are individual estimates that you can use as covariates in your models, and also principal components, which is based on using genetic data and parsing out variants, and then those principal components then are assigned. Each person will have principal components that explain the variance, and you can use those as covariates. Multidimensional scaling is similar, except that instead of variance, you're looking at sharing, and you're creating the same sort of components that can be used also to adjust in a model. So we'll talk about this again, but this audio helps us wrap up this lecture. And if you have any questions, we can talk about this later. Thanks.